Hi, welcome to our channel of IGNOU audiobooks, Indira Gandhi National Open University, School of Sciences, SOS, Bachelor's degree programs. Bachelor of Science, Honours, in Biochemistry, BSCBCH. Core courses. BBCCT-101 Molecules of Life. Block-1 Introduction to Biochemistry. Unit-1 Foundations of Biochemistry. 1.1 Introduction. The term biochemistry is self-explanatory, which is evolved from two basic science subjects, i.e., biology and chemistry. Biochemistry is a branch of science that deals with chemistry of life and living systems. In your earlier classes you might have studied both biology and chemistry as individual subjects. In this unit, you will see how biology and chemistry merged with each other, leading to the genesis of biochemistry. You shall be introduced to the significance of biochemistry in understanding our health and disease status. You will be motivated by studying and understanding the cellular and molecular basis of life in a cell. You shall also learn how various chemical entities involved in maintaining life inside biological systems. Much of life can be understood in rational terms if expressed in the language of chemistry. It is an international language, a language for all of the time, and a language that explains where we came from, what we are, and the physical world. Will allow us to go. Arthur Kornberg, 1918 to 2007. Besides this, you will be briefly introduced to the major historical discoveries occurred in biochemistry that enabled human beings for better understanding of the chemistry of life. Along with the glorious history, we shall also describe how biochemistry progressed as an interdisciplinary branch of science. The best part of biochemistry is, it includes the basic principles of all major science subjects and answers the unanswered questions of life in living systems. At the end, we shall also discuss units of measurement used to express various physicochemical properties of substances. 1.2 Origin of biochemistry. Every mother who curdles milk or prepares dosa dough is a biochemist. It may sound little ironical, but it is true. Human beings have been using the applications of biochemistry like fermentation, pickling, and tanning since ages. But they were not aware of science behind it. In the early 19th century, the path-breaking work done by Friedrich Wöhler, a German chemist, laid the stone, which further directed to the development of a new branch of science that is recognized as biochemistry. The distinguishing work done by Wöhler is the synthesis of urea in the laboratory without involving any living, biological material. He did this pioneering work in the year 1828 by heating ammonium cyanate. The interesting part of this synthesis was production of organic substance from inorganic substance. NH4 is equal to NCO yields with heat H2N bond C bond NH2. Another important milestone in the history of biochemistry was the work done by Louis Pasteur, where he explained the significance of yeast cells in converting sugar to alcohol. This landmark work was performed in the mid-19th century. Pasteur explained that yeast cells encourage fermentation, alcohol production process. He also established the fact that bacteria present in the fermentation mixture are responsible for the production of citric acid, sour taste. Since then people started avoiding usage of bacteria in fermentation mixtures. This finding led to a major breakthrough in the process of wine production. Later, in the year 1897, Eduard and Hans Buchner, the two German brothers, showed extracts of yeast cells could carry out the entire process of fermentation. Buchner's work disclosed the possibilities for in vitro, Latin, in glass, research. Experiments that performed inside a test tube or outside a living system are called in vitro experiments. However, the experiments carried out in a living system are referred as in vivo. Later, in the year 1903, Carl Newberg coined the term biochemistry. In the first half of 20th century, some of the basic reactions occurring inside biological systems were explained. One of the significant metabolic cycle was illustrated in 1937 by Hans Adolf Krebs. He described citric acid cycle for which he received Nobel Prize in Physiology. Similarly, the reactions of glycolysis, splitting of sugar, were explained by Gustav Embden and Otto Mayhoff in 1940. These are some of the significant discoveries that showed a wonderful impact in the field of biochemistry. Up to mid-20th century, it was believed that proteins are hereditary factors. But the experiments conducted by Hershey and Chase in 1952 proved that DNA is the genetic material but not proteins. Later in the year 1953, groundbreaking discovery of the 20th century took place when James Watson and Francis Crick described the double helical structure of DNA. This discovery helped science fraternity to understand how genetic information is stored in DNA. In the year 1968, Hargobind Khorana, an Indian-American biochemist, discovered genetic code and received Nobel Prize in Physiology in 1968. Major progress in biochemistry took place in mid-20th century due to the development of several biochemical and biophysical techniques like chromatography, NMR, X-ray diffraction, radioisotope labeling and electron microscopy. This journey of biochemistry is continuing and expanding every day. With the inception of cloning, genetic engineering and several other advanced molecular and cell biology techniques. The major discoveries that happened in the fields of chemistry, physics, biology and advanced cell biology allowed scientists for the better understanding of life and its chemical existence. As time progressed, biochemistry evolved as an independent subject with interdisciplinary concepts, components and connections among basic sciences, biology, chemistry and physics. Hence, there is no hesitation in saying that biochemistry is an interdisciplinary subject in its nature. In next section, we will be studying more about the interdisciplinary nature of biochemistry and how and why biochemistry is considered as an interdisciplinary subject. 1.3 Biochemistry as a discipline and interdisciplinary subject. So far we have discussed about historical background of biochemistry, during which we came to know that biochemistry appeared as a central part of research interest in chemistry as well as biology. Apart from these two subjects, biochemistry interacts with fundamentals of physics and computer science, Fig.1.1. Now, let us discuss in detail about interdisciplinary nature of biochemistry by taking few examples and applications of core research areas. In biochemistry, structural biochemistry, in this area of study major inputs will be taken from basics of organic along with physical chemistry. This area of study is useful in understanding how and what composes life. In this, we will be studying about biomolecules, structures, functions and their interactions with each other along with thermodynamics of a living system. Biophysics. This is another widely studied area of biochemistry, where researchers will be interested in knowing the kind of physical interactions taking place between biomolecules and the surrounding environment. These interactions may be either inside the cell or outside the cell. That enables the maintenance of life in a living system. Most of the biophysical techniques will be discussed in detail in course BBCCT-105. Proteins depend on basic physical and chemical properties of biomolecules. These techniques are frequently used in diagnostics, medical and clinical research fields for separation, isolation, purification and identification purposes. Cell and molecular biology. This is another upcoming area in biochemistry research with wide opportunities for discovery and innovative research. The major principles applied in this field are from microscopy, electron, confocal and fluorescence microscopy, microbiology, cell and tissue culture. Medical and clinical biochemistry. It is interesting to note that all medical graduates and paramedical students read biochemistry as a compulsory subject. The reason why we are emphasizing this point is to understand health and disease status of any living system. Knowledge of biochemistry is very much essential. In simple words, any imbalance in the normal metabolism leads to metabolic disorders causing disease. Fig.1.2. The one with sound knowledge of biochemistry can easily identify and understand the cause of disease status. Fig.1.2. This diagram shows how knowledge of biochemistry is useful in understanding the health and disease status of an individual. For example, detailed knowledge of lipid metabolism is needed to address the issues related to obesity. Nutritional biochemistry. This is another junction where biochemistry crosstalk with principles of nutrition. Our diet is majorly composed of carbohydrates, proteins, lipids and vitamins. These are the major biomolecules that are responsible for energy, structure and functioning of our body. Hence, one with adequate knowledge of biochemistry is capable of understanding reasons for certain dietary disorders like malnutrition, kwashiorkor and marasmus. Moreover, lifestyle disorders like obesity and diabetes are well managed by the sound knowledge of biochemistry. Bioinformatics, other upcoming and promising area which is widely involved in drug discovery and design. In this area, by using computers, biological data can be accessed, stored and retrieved. Tools of bioinformatics are none other than bioinformatics softwares and high-end computers, which are developed by using principles of maths, physics and chemistry. The major applications of bioinformatics are in the field of biochemistry and pharmacy. These are some of the important subareas of biochemistry which clearly indicates that biochemistry is having interactions with almost all fields of sciences. Hence biochemistry evolved as an independent branch with interdisciplinary nature. 1.4 Cellular and chemical foundations of life, chemical elements of cells and living systems. Up to now, we have discussed interdisciplinary nature of biochemistry. Now it's time to know the basics of life, i.e., in which form does life exists. In your lower classes, you might have studied that cell is the basic unit of life in a living system. The hierarchy of organization in a living system is like cells to tissue, tissues to organ, organs to a system, and systems to an organism. The complexity of organization and structures slightly varies. From invertebrates to vertebrates, in other words, from smaller organisms to higher organisms. At the same time, the composition of the cell varies. From organism to organism. Fig. 1.3 illustrates the chemical composition of a normal human. We all know that on this earth life exists in different forms like microorganisms, plants, and animals. All these forms of life display certain common properties, like motility, except plants, growth, reproduction. The fact to be remembered here is that all these organisms are made up of similar organic and inorganic chemical entities like proteins, carbohydrates, lipids, nucleic acids, and vital minerals. The crux to recognize in this section is these biomolecules are common in their basic composition, precursor elements like carbon, nitrogen, oxygen, phosphorus, etc. Refer Table 1.1, but differ in their arrangement from one organism to another. Table 1.1, Inorganic elements and their biological functions. Carbon, C, hydrogen, H, oxygen, O, nitrogen, N, synthesis of organic compounds like proteins, carbohydrates, lipids and nucleic acids. Sulfur, S, a component of sulfur-containing amino acids. Sodium, Na, regulates a osmotic pressure in cells. Helps in the transmission of nerve impulses. Magnesium, Mg, acts as a cofactor of kinase enzymes involved in protein synthesis. Calcium, Ca, required for the formation of strong bones and teeth. Helps in muscle contraction. Promotes blood clotting. Phosphorus, P, synthesis of nucleotides, synthesis of ATP. Formation of strong bones and teeth. Chlorine, Ci, component of the biological buffer. Synthesis of hydrochloric acid by the gastric glands in the stomach. Potassium, K, regulates osmotic pressure in cells. Required in muscle contraction and transmission of nerve impulses. Copper, Co, cofactor for several enzymes. Iodine, I, involved in the synthesis of thyroid hormones. Ferrum, Fe, involved in the synthesis of hemoglobin and respiratory enzymes. After going through above table, we need to realize that forms, shape or size of biomolecules may be different but their basic composition remains same. Let us understand this concept in detail. Non-living to living. It might be surprising to know that major biomolecules are synthesized from non-living inorganic chemical elements. When we observe these non-living elements, they exhibit all the physical and chemical properties that are characteristic of a non-living or lifeless material. But these lifeless elements arrange to form biomolecules, Fig. 1.4, like carbohydrates, proteins, nucleic acids and lipids. Their precise interaction with each other is responsible for the maintenance of life inside a cell. Fig. 1.4, Synthesis of biomolecules from inorganic elements. Look at the screen. In your lower classes you might have studied that DNA acts as a hereditary genetic material that carries information from one generation to the next. Both animal and plant cells possess structurally similar DNA, double helix, i.e., composed of structurally similar nitrogenous bases like adenine, guanine, cytosine, and thymidine. Though these DNAs are structurally similar, they differ in their functions, expression of characters. Here we need to understand that In spite of structural similarities in DNA double helix and nitrogenous base composition, they differ in their nucleotide sequence, that is responsible for functional diversity. In nature, every living cell is capable of synthesizing desired biomolecule according to the genetic information available in its DNA sequence. In the process of biological synthesis of biomolecules, enzyme machinery present in a living cell uses chemical elements as precursors. Since from the beginning of this section we have been studying that biomolecules are vital for maintenance of life, let us know some of the important biological functions exhibited by biomolecules in Table 1.2. You will be studying in detail about the structure, classification and biological significance of vital biomolecules in next units of this course. Important functions of water, inorganic molecule, in maintaining life are acts as solvent, major component of body fluids, regulates body temperature, vital molecule in digestion, absorption and respiration. Table 1.2 Important functions of biomolecules. Biomolecules and their functions. Carbohydrates primary source of energy. Proteins form structural components like myosin of muscles and keratin of hair. Build new cells for growth and renew damaged tissue. Involved in the synthesis of blood clotting factors and hemoglobin. All enzymes are proteins. Lipids source of energy. Phospholipids are major constituents of the plasma membrane. Maintenance of body temperature. Synthesis of wax. Nucleic acids hereditary factor. In this picture a toy house is made by using blocks. You can dismantle and rearrange these blocks to form another shape or toy model. Imagine these blocks with elements like C, O, N, H, S, P etc. that rearrange inside living cells to form desired biomolecules. 1.5 Units of measurement in biochemistry. Units of any measurement play a vital role in our routine life, starting from using milk in kitchen to purchasing a designer-made suit. These units of measurement facilitate scientists across the world to communicate and exchange their results with one another. The units related to scientific measurement are assigned by the International System of Units, SI. SI is the short form of the French term Les Systèmes Internationaux d'Unités, developed in 1960 from an older metric system. In this section you will be studying about different types of units used to quantify or for standard measurement of various materials and reagents used in biochemistry. Biochemists routinely use units such as microgram, milligram, liter etc. to express the results in a biochemical analysis. These units enable to observe and analyze the data obtained before and after conducting experiments. Table 1. 3. AC shows some of the basic units like weight, length and volume used frequently in practical biochemistry. However, we will be discussing the detailed terminology of specific concentration like molarity, m, and normality, n, in the lab course, BBCCL-102. Biomolecular dimensions, the dimensions of mass and length for biomolecules are expressed in Dalton and nanometer. Molecular mass is denoted as m. This is defined as the mass of one molecule. 1. Dalton, Da, is equivalent to 1/12 the mass of carbon, a kilodalton. Kda is 1000 daltons, a megadalton. Mda is 1 million daltons. However, nanometer is abbreviated as nm. 1 nm is equal to 1 billionth of meter, i.e., 10 to 9 meter. Refer length in Table 1.3. 1.6 Summary. Origin of biochemistry took place early in the 18th century. Later in the mid of 19th century, biochemistry established as a subject. The word biochemistry was first proposed in 1903 by Carl Neuberg, a German chemist. Biochemistry has become the language of biology and medicine. Modern biochemistry evolved on the applications of advanced techniques to biological problems. Biochemistry deals with the structural and functional chemistry of the living organisms, that include the structure of individual molecule and their molecular regulation and expression. Owing to the interactive nature of biochemistry with various branches of science subjects, it is considered as an interdisciplinary subject in nature. It is interesting to note that biomolecules are composed of inorganic elements like C, N, H, O, S, and P. The basic unit of life is cell, which further joins to form tissue and so on to form a complete organism. SI units are used to measure and express various properties of biomolecules. Thank you. Subscribe to our channel for more updates, and we will see you with the next chapter.